Hi, so this is my first proper tutorial revision video sort of thing, so yeah, go easy if I kind of suck, but anyway, right. So this video is a requested one by a friend of mine, Charlene Bell, and it's on biology, specifically diffusion, and I will just go through membranes and membranes in relation to permeability. Then I will just a quick definition of diffusion, and then I'll go on to a definition of osmosis. So after that, then just quickly go through the mandatory experiment, which is to demonstrate osmosis. And then I think I'll just go through osmosis in cells, in animal cells, and then in plant cells, just what happens, that sort of thing. Yeah, so I'll just get oh yeah, membranes. Membranes, one thing to remember is that for the most part, all biological membranes in a cell are all the same in that their structure is the same, and they tend to work the same, as in one rule fits all kind of thing. So the membrane that's around the cell itself will be the same kind of membrane that's around the mitochondria and the chloroplast in the plant cell. Okay, so there are three types of membranes. There are permeable membranes, which is just everything goes through. There's impermeable, nothing goes through. And then there's selective or semi-permeable. And obviously, as the name implies, some things go through and some things don't. So, for example, it could allow water molecules to pass through, and then it wouldn't allow sugar molecules to pass through, glucose molecules. That's basically it. And then moving on to diffusion now. A good definition of diffusion, a good handy definition, is just it is the movement of molecules from an area of high concentration to an area of low concentration. So a good way to remember that is that in a biology classroom, if somebody opens the gas tap on the other side of the room, eventually you're going to smell it if you're on the opposite end of the room. That's just basically it was over there, high concentration, or over where you were, it was low concentration, and eventually it moved over. That's really it. That's all you need to know, basically. And then examples of diffusion in biology would be carbon dioxide diffusing into the leaf. It's obviously through the stomata. And then there's oxygen diffusing from the blood. Anyway, so osmosis. Sometimes I'll ask a question on osmosis that's kind of like, why is it described as a special case of diffusion? And that's just that it's kind of like diffusion, only specifically with water. That's just it, because diffusion is the movement of all gases and all liquids all together, everything. But then osmosis is just diffusion specifically in only water. That's all. But then the proper definition of osmosis is the movement of water molecules from an area of through a semi-permeable membrane. That's an important thing to remember. So through a membrane from an area of high concentration to low concentration. And the area of high concentration, we call that hypotonic, and an area of low concentration of water, we call that hypertonic. So hypo, that'd be H-Y-P-O, tonic, and then hyper. A good memory key, I kind of think, is hyper is usually sugar or salt. If you want to remember it through sugar, sugar makes people hyper. No, works for me. I don't know if it worked for you. But anyway. So then, that's basically on the definition. That's basically it on the definition itself. The theory of osmosis is just basically, if you have on this side pure or distilled water, whatever, that's not a solution of anything, just water. And on this side you have salt or sugar solution. The solution, by the way, is just a mixture of a solute and a solvent. Solvent being water, solute being sugar or salt. So a solution anyway, so a salty sugar solution this side, pure water on this side, and a semi-permeable membrane in between, which remember I said allows some things, like water for example, and not allow something like sugar. So the water will move from the area of high concentration, which we said was hypotonic, to the area of low concentration, which we said was hypertonic. And that is osmosis. That's really it. Another thing to mention is in a situation where the semi-permeable membrane is dividing areas of equal concentration of water, that is called an isotonic solution. So that would be, good way to remember this that my teacher told me was that when they're about to do a heart transplant, when they take it out of the donor, they put it into an isotonic solution so that the muscle won't shrink because water will move out of it, and so that it won't burst because water moving into it. So it's equal. So that means there's equal movement of water in and out. So nothing explodes and nothing shrinks. So that's called isotonic. Yeah, so then we'll just quickly go through the experiment to demonstrate osmosis. So it's just basically you have two bowls or whatever. I actually can't remember the proper name for them, but you know, the sciencey looking bowls that you put water in. So you have two filled with water, and you get your Viscon tube, and you get two because you're going to obviously need a control as well as an experiment. In one of them you have sucrose solution, and in one of them, your control, you just have water, plain water. You tie it to something that you can use to hold it on top of the bowl. You can't just like actually sit it in there, so you can tie it to a pencil. I think I can show you the picture that I have here, just in case you're not getting this, but this is something you should know anyway. If you don't, kind of like, sorry, a bit lower. No, okay, sorry, I have really bad hand-eye coordination. There we go. You see that? Like, there's a pencil kind of thing, and there it is hanging. And there is your control. That one right there, that's your control, and that's your experiment. And is you leave it there. The one with the sugar solution is only 80%. It does have to be some water in it, obviously. You leave it there for a little while. When you come back, the one with the sucrose solution, you're going to find out that it becomes turgid. Turgid just means stiff, basically, because it's filled up with water. Now it'll stand upright, like the way we had our when we did it, like when we actually did the experiment in class. The way we had ours was that we had something to kind of hold it limping, or not limping, it's not the word, drooping over the side of the bowl, so that when it became turgid, it actually stood upright, kind of in the bowl. So it just made it that bit more obvious to see the difference. That's kind of it. I may or may not, I probably actually won't type up this experiment in the blog, because if you're doing biology at this stage, I'm pretty sure you have a textbook and it's in there. That's just a basic breakdown of it in layman terms for you, in case you didn't get it when your teacher was going on about it. Okay, so now we're going to go into osmosis in animal cells and plant cells.